Hi, welcome to this lecture on mathematical methods for biologists. We learned about diffusion and we learned that how to use calculus to understand the diffusion and the diffusion equation. Today we will learn one more thing about, we will try to learn to calculate some things from the equations that we learned, which would be very useful in biology, and to estimate quantities about like how far a particular protein will diffuse and so on and so forth. And today's topic therefore is diffusion versus active transport. So that's what we will discuss today. And the question we will try to answer in this lecture is how far will a molecule diffuse? And what is the difference between diffusion and active transport? So this is something that we will discuss. We will try to answer these two questions, that how far will a protein molecule diffuse in a typical condition, and what is the difference between diffusion and active transport. So what we learned is an equation which describes the diffusion of protein monomers. So, and the equation we described was that if C is the concentration of protein molecules as a function of x and time. Instead of x, it would be x, y, z. If we know this, if we want to know the concentration as a function of x and t, if we know some quantities, this C can be obtained by solving this equation del C by del t is equal to d del square C by del x square. So this is the equation that we learned for this C. This is the diffusion equation, and by solving this equation, we will get C as a function of time. So this C is a solution of this equation. So to get C as a function of x comma t, we have to solve this equation. And what we know is that to solve any differential equation, we need something called initial condition. So here there is a derivative in time, therefore we need some initial condition. And there is a two derivatives in x, therefore we would need two conditions for that, which we would call typically boundary conditions. So we would need two boundary conditions to solve such one initial condition and two boundary condition. If we provide this, we would be able to solve it. So let us think of the following situation, which is important to understand this. Imagine that you have a cell, as we have been imagining, a long one-dimensional cell which is very long, and if you imagine that time is equal to 0, so time is equal to 0, we assume that all the protein molecules are just one point here. So only at this particular point we have protein molecules only here. So if I plot concentration versus x at t equal to 0, this would be almost like a delta function. Almost will be a function only has value here, everywhere else the function is 0. Such functions are called delta functions, Dirac delta functions. If you want to assume this is you can even imagine this is a Gaussian with a very small width. So you could imagine this is a Gaussian with a very small width. So you can imagine this is a Gaussian with very small width if you want. So you essentially have a concentration which has a peak essentially at one point. Now after 10 minutes, if we look at this, so when you look at t is equal to 10 minutes, how will that concentration versus x will appear? It turns out that as the time progresses, this would diffuse this way and this would diffuse this way. And the concentration at this x values, for example, the concentration here and the concentration here would be slightly higher. It will be non-zero. Therefore, after a long time, after sufficiently long time, you would get some, so this would be this, you would expect some function something like this. Some symmetric function, something like this. This should be symmetric on either side. I can draw this better. A symmetric nice function like this. Now, let us imagine this further. So, if I start with t equal to 0, we have a delta function, and as time goes, it would be a function like this, and as it goes further, it will spread further. So, let me draw this. It would spread further and further and further. So, you could imagine concentration versus x, concentration versus x, concentration versus x. So, this is t equal to 0. So, this is equal to t is equal to 10 minutes. This is further like t is equal to 20 minutes or 30 minutes. So, as the time progresses, the concentration would have a functional form, something like this, naively speaking. Now, mathematically, how do we get it? We can get this by solving this equation for appropriate boundary conditions and initial conditions. Depending on various boundary conditions, initial conditions, one will have various solutions. This need not be the solution always. This is a typical solution. So, such solutions are obtained by assuming two things. One is at t equal to 0, the concentration is highly peaked at one point. The concentration is a delta function, and as time at far away, that is here at this boundaries, like if far away here, the derivatives and concentrations are 0, and assuming this kind of conditions, at infinities the derivative and the concentration is 0, and at t equal to 0 it has a delta function. If you assume such boundary conditions, one will get nice symmetric functions. So, a symmetric function which represents this is a Gaussian function. So, a typical solution for this kind of a diffusion equation is a Gaussian function, which is c of x comma t. So, if you assume nice initial condition and boundary condition, one would get a typical solution which you would read in textbook. Which has some functional form like this: e power minus x minus x0 whole square divided by 4 dt at x equal to x0, which is this location. So, this is my x0. So, at x equal to x0, you will have a peak and it will be symmetric around x0, and as time goes, this denominator term will increase, and which would make this spread. So, this is the solution for the diffusion equation under some particular initial and boundary conditions. So, I urge you to read about it, learn this a little bit more, but does not matter. What you should know is that by solving those equations, one can get a c as a function of x comma t, and one would be able to know the concentration at any point. What we are further interested is the following. Imagine that you have a cell, like if you imagine a cell. At t equal to 0, as we just said, there is the protein molecules are only at one point. So, this is at t equal to 0. As the time progresses, this would spread, as we know. So, I do one experiment. Let us say I did one experiment and I saw that if I focus, so I have this situation, I will focus on one molecule. So, let us focus on one protein molecule, and if I track this protein molecule, I would see that this is going in some random direction. So, let us say it went in this direction first and then it took various random directions. In some other experiment, if I repeat the experiment, exactly under same conditions, I repeated exactly same experiment, same condition, and I tracked another molecule. This time this might as well go this way, we don't know. So, on an average, if you average over many experiments, it is a fair question to ask, what is the average position of particles of a given particle as the time proceeds or at any time? And it turns out that if you average over many realizations, since things are symmetric, the x average will be x0, or if x0 is 0, the x average will be 0. So x average would be 0 if x0 is 0. Otherwise, on an average, it would be at the peak at this initial position itself. It would as if that on an average the position would be the x0, which is the initial position itself. Now, how much would it spread? How much is this spread? This is calculated, how much it would, at any given time at 20 minutes, how much it would spread. This spread is basically calculated by this quantity called root mean square distance, as we would learn a little bit in the statistics, or anybody who has learned statistics would know that the spread, which is x square average minus x average square. This is the spread, which is the standard, which is the variance. So this is the variance, and this is basically tells us that how much would a molecule spread after some time. The amount of spread, the width of this distribution is essentially given by this, and it turns out that this quantity, which let me call sigma square, which is x square average minus x average square, and it turns out that this is equal to, in one dimension, it is 2 dt. So x square average is 2 dt. If x average is 0, if I can take x0 equal to 0, so then it says x square average is 2 dt. So interesting to note that the square of space variable x square is proportional to time. This is important to remember. This is a very important property of diffusion, that x square average, so this angular bracket would mean mean or average. Mean, average, this is the angular bracket means this is equal to 2 dt in one dimension, and in 3 dimensions it will be 6 dt, in 2 dimensions it will be 4 dt. So x square average is 2 dt. This is also sometimes written as x square bar. Bar would also mean average or mean. So this is 2 dt. It's important to remember the square is proportional to time. The root of this, which is called the XRMS, the RMS distance. So this is, let me call this XRMS, root mean square distance. So I find root of mean and square. In other words, some place it will be written as x square bar like this, which would of course be root of this. It will be root of 2 dt, square root of 2 dt. So this is an important relation that one should remember to calculate the spread, how far the protein would, the concentration would spread if I start from one point. So using this, one can estimate the distance, a typical distance, the root mean square distance is the typical distance. Typical distance protein would spread given some time t. This is answered by this formula. So let us calculate this XRMS, which, so let me physically explain this once more. This is very important for all of you to understand. If we imagine a cell as we have been imagining, and if we consider at t equals 0, a lot of proteins accumulated at one point, and as the time progresses, this protein would spread. So let us say this is after 10 minutes, the protein has spread to a larger distance. So you have few here, and this has spread to a larger distance. How far this spread is? So this distance over which this has approximately spread. This is the physical measure of XRMS, which is root of 2 dt. This is the root mean square distance in one dimension. This is 2 dt. In 2 dimension it is 4 dt, and 3 dimension it is 6 dt. So this let us compute this spread, the distance over which a protein would spread, given a time and a diffusion coefficient. We can compute this, and let us do this for a particular protein. So let us assume these are protein molecules having a size of 1 nanometer. So let us assume protein having diameter 1 nanometer at 300 Kelvin, and let us assume viscosity of water which is 10 power minus 3 SI unit. So then we will ask the question, how much it will spread? What is the spread can we measure? And this spread is calculated by this. So let us calculate this 2 dt. So in 1 second, if I take t is equal to 1 second, root 2d will give me the distance, the spread over 1 second. So root 2d will give me the distance spread over 1 second. So let us do that, calculate here. So let me calculate x square. So d for a protein is kbt divided by 6 pi eta a, and if I substitute these values, this will be approximately 10 power minus 10 meter square per second. Root 2d, and if I take 1 second as the time, this will be root 2 into 10 power minus 10. So this is approximately 10 power minus 5. 10 power minus 5 SI unit is meter. So this is going to be the XRMS. So in other words, this is approximately 10 micrometer. So in 1 second, the spread would be approximately 10 micrometer. So this is an interesting answer that we would get, that if I take a 1 nanometer protein and ask the question that if I take a concentration of 1 nanometer protein and ask the question how far it will spread in 1 second, the answer is approximately 10 micrometer. So this is one interesting point. Another interesting point one should remember is that let us consider 1 second and 100 seconds. So let us do this. XRMS t is equal to 1 second. We found this is 10 micrometer. Now so this is t is equal to 1 second. If t is equal to 100 second, XRMS will be root 2d 100, root t is 100. So this is approximately equal to 2 times 10 power minus 10 times 100, which is 10 power 2. So this would be approximately root 2 times 10 power minus 8. So this is, let me approximately write it as 10 power minus 4, and this is basically 100 micrometer. So this is approximately 100 micrometer. So in 1 second, it was 10 micrometer. When the time was increased by 100 times, so the time was increased from 1 to 100 times, the distance increased by 10 times. So the x is increases like square root of t. This is the hallmark of diffusion. When I increase time by 100 times, the x only increases by 10 times. It changed from 10 to 100, 1 to 100 here. Let me say this. If I take protein 1 nanometer size, if I take this protein and ask the question how far it will diffuse in 1 second, 10 micrometers answer. In 100 seconds, 100 micrometer. So the time was increased by 100 times. So the time 100 times. The distance, so this is time. This is the distance spread. This increased by 10 times. In other words, the distance variable x, the spread, is proportional to square root of time. This is the hallmark of diffusion, which all of you should remember. If I increase the time by t times, n times, this will increase only root n times. So that is the hallmark of diffusion that all of you should remember. Now, as opposed to diffusion, there is active transport inside the cell. For example, there are molecular motors which would carry and transport things. So if you consider a cell, there are two kinds of transport one can imagine. So you have a cell and there are molecular machines which would carry particles, vesicles along a microtubule. So there is an active transport. So this are called molecular motors. These molecular motors will transport using ATP. They will use ATP and transport things. And how far will this go in as a function of time? So these molecular motors have a velocity. So velocity of molecular motors has a velocity. So velocity v for molecular motor. Molecular motors have a velocity. Velocity is distance by time, right? It is like x by t. So x is vt. So here in the active transport, so this is transport by molecular motors are called active transport. So let me call this active transport. What do I mean by active transport? This is transport by molecular machines. So they apply some force and carry something. So there is a force typically applied. They use energy from ATP hydrolysis or the food we eat, and it transports things. So there are typical transport molecular motors move along microtubule. The typical molecular motor names are kinesins and dynein. So they walk along microtubules and transport vesicles. This should be contrasted with diffusive transport. So here just remember x goes as vt. There x went like root 2 dt. So let me contrast this transport, active transport versus diffusive transport. So in the case of diffusive transport, so this is diffusive transport of protein. What we learned is x is proportional to root time. This is the thing that we learned. And in the active transport, where the transport happens because of either some external force or molecular motors, x is proportional to t. So this is the difference. So if I increase this by 100 times, this would increase only by 10 times the distance traveled. If I have molecular active transport, if I increase this by 10 times, this would increase also by 10 times. If I increase this by 100 times, this would also increase by 100 times. So this in the active transport, the distance traveled x is proportional to time. Here the distance traveled is proportional to square root of time. This is an important difference that all of you should keep in mind. And one interesting thing that all of you should do is use this idea and calculate the time it takes for a particle, a protein of your interest, to move for example, across a long neuronal cells, how long it would take, what is the time it takes. I urge all of you to calculate. You would see that diffusion is very slow, and what would need an active transport to take things far away. So now the interesting thing here, another thing to remember is that in one case the x, the with 2 dt, and the d is controlled by temperature, which is a fixed quantity in a cellular environment. Viscosity is also kind of fixed. The size of the protein is also fixed. So this is not at all in our control. D is not at all in control. So if I have 1 hour time, this can only go that much. Sorry, x square here, x square. So x is root 2 dt. The x is root 2 dt. Since d is a constant given a biological environment, this can only go this much. On the other hand, the velocity, which is the active transport, x is vt. Here the velocity is not really constrained. So if there is an external force, we saw that this is F by 6 pi eta a. So as I increase the external force, I can increase the velocity, or I could drive this by hydrolyzing ATP. So the more ATP I consume, I can drive it faster. So this velocity only depends on the availability of ATP and so on and so forth. So I can move much faster using the active transport as opposed to the diffusive transport. So it is important to understand this difference between active transport and the diffusive transport. And this gives us some basic details about these two different types of transport and the mathematics behind this. So with this, I will stop this lecture and continue in the next lecture. Bye.